Hi, my name is Shaddy and I'm here today to tell you about my journey. As a kid, I was always curious to know about how our bodies work and interested in understanding the biology of living organisms. And that was the base for me to start and study bachelor degree in biotechnology and genetic engineering, followed by a master degree in applied biological sciences from Jordan University of Science and Technology. After that, I was working for long years in academia and research, but still, the kid inside me wanted to explore more and understand more. Then, following that, I won a full scholarship to continue my PhD study at Weizmann Institute of Science, one of the world's top leading research institutions. And there, I gained the interest to understand stem cell biology and mammalian development. Mammalian development occurs inside the uterus, and the uterus barrier blocks us as a scientist to observe and understand these fascinating developmental processes. And for that, the question was for years, are we as a scientist, we will be able one day to grow a mammalian embryo outside the uterus? If this will happen, it will be a revolution in the modern biology and will allow us to observe more and understand more not only developmental process, but developmental errors also. In 2020, I was part of a research team that after 7 years of hard work and preparation, we were able to create the world's first artificial womb, or what we called ex utero culture system, that contain an incubator, a gas mixer, and a rotating drum that allow us precisely control culture condition from temperature, oxygen, pressure levels, and by using our highly enriched medium, we were able to grow natural mouse embryos from early stages, what we call the pre-gastrulation, to later stages of organogenesis. All of this outside the uterus in the lab. This revolutionary breakthrough discovery was taken by Nature for publication, which is one of the top research journals, and was highlighted in many news, like New York Times, Science News, MIT Technology Reviews. All of these news were talking about this kind of discovery. Not only this, in 2021, it's considered as one of the major breakthroughs in science, as considered by Science magazine. Following this kind of discovery, we were asking that we now succeed to take mouse embryos from the uterus and culture them for 6 days outside the uterus. And now we shift to ask, what about stem cells? How we can take just a stem cell on a plate in the lab and try to culture this kind of stem cells and reach later stages. The big dream for every stem cell biologist that we will be able one day to take a patient's skin cell, revert it back to its origin, and what we call it induce pluripotent stem cell, and use this stem cell as a model to understand the early pathology and the early error, what is happening there. Imagine in another situation, in abortion, when most of the couples don't know even the reason or what went wrong. If we can take a cell and revert it back to the origin and use it as a model to follow back and capture all the developmental process and try to understand what is happening there. Still, this is a dream. And despite all of the progress that have been done so far in the science field, no one was able to take this kind of cell and model advanced later stages of embryogenesis. I decided to take this challenge in my PhD because I thought that it should ideally work first, and if it will work, it will be a revolution that will reduce the use of animals and will open a new era in translational medicine field. So I started by engineering cells, stem cells in a plate, and mix all these cells together. And by using of our technology, this ex utero culture platform, and after a long way of precise trials and errors. Guess what? It works. I was the first one to create the first synthetic mouse embryo model, just from stem cells. These embryos were able to reach later stages with heart beating develop, with the brain, with blood flow. This breakthrough discovery was taken by Science, by Cell, which is one of the major journals inside the field, and it will get recognized by a lot of news agencies like The Guardian, Washington Post, many other news agencies. Not only that, but get translated to different other languages and was highlighting the news in India, Japan, Germany, Italy, China, and others. This milestone, breaking through discovery, will be a valuable tool to understand early development and try model genetic disorder and try to do drug screens and apply organ transplantation. To be clear, we are not aiming to do alive embryos out of this model, but we want to use it to understand how is the development happen and how is the organ form. Simply, the embryos like 3D bioprinter. It's the best to make organ, and we want just to learn from it how to do it. This kind of research is taking a lot of time, there is a lot of difficulties, but it's opening a new hope for the big dream that I told you in the beginning to be one step closer. Actually, a Renewal Bio, which is a newly established biotechnology company by my advisor Yaqub Hanna, that aiming to take this kind of technology, ex utero synthetic embryology technology, and applying using human stem cells, try to push human health and try to solve many human issues like infertility. Success is not easy. It's a long way full of difficulties. But you must believe in your dream and keep fighting for it. Even if no one sees it for you, you should see it for yourself. You deserve the best that you see for yourself. I am one story that have a chance to be here today and you are listening to it. But there are additional 2 million stories in Gaza that they only need a chance to do the impossible. Thank you.